Everyone. I'm Aaron Noel, along with my co-host today, Audrey Chaton. We're here representing the Network for Ocean Worlds in the quarterly lecture series. This season is about making measurements on ocean worlds, and and episode 3 is on making and utilizing measurements of organics on Earth and ocean worlds. We have two great speakers today, Dr. Dennis Hansell and Dr. Fernanda Mora, and give you a little more intro on them as we come to each of their talks. Without sort of further ado, let's get going with the first half of our talk, which will be focusing on Earth. And so our speaker is Professor Dennis Hansell from the University of Miami, the Rosenstiel School. He's also a fellow of the AGU and a fellow of AAAS, and he has a significant expertise in the Earth's ocean and the dissolved organic cycling there. And he'll be giving us a talk today on illuminating the role of dissolved organic matter in the global ocean carbon cycle. So without further ado, Dennis, please take it away.
Thank you so much. Good to see good turnout here. Appreciate that. And a couple of familiar names and friends in the audience. That's good too. Let me see if I can get this going. Okay. All right. Anyway, thank you. I apparently didn't remember the title fully, so I made up a new one here, which is basically the same. I want to, again, thank NSF, NOAA, and NASA for support over the years. I've been in this doing the questions about dissolved organic matters, but particularly carbon in the global ocean for a long time. And they have been steadfast in their support and, greatly appreciate that. I put the image here because, a lot of the data I'm going to show you looks really static and quiet and all that sort of thing. But this is what it's more commonly like for people out there, suffering and sampling for us, because our cruises are long. They could be 5 weeks, 6 weeks, 7 weeks, 2 months, that sort of thing. And often they can be very rough. So I'm going to, talk about dissolved organic carbon in the ocean, but I'm going to try to, towards the end, look for parallels, from a very naive perspective between Earth and, the world's other ocean worlds, let me say that. Okay, so if you want to learn more about dissolved organic matter in the ocean, Craig Carlson and I have we edit this book. It's coming out in the 3rd edition in July of 2024. And you can learn about composition, microbial dynamics, optical properties, sediments, hydrothermal involvement, organic nutrients, photochemistry, volatiles, long-term stability, role of rivers, global dynamics, modeling, that sort of thing. So if you want to learn more, there's that book coming out, and then there's obviously 2 prior editions with some other chapters in them. Okay, so today I was asked to talk about measurements a little bit, how we do it. So determining DOC concentrations, and doing surveys of the global ocean is what I use mostly to figure out what's going on out there. And when we do those surveys, we're surveying. Here's a schematic of kind of a ribbon diagram of ocean circulation, with the reds being at the surface and those greens sort of being intermediate waters and the dark colors being deeper in the water column. And you can see there's a lot of complexity in the system. You can't measure dissolved organic matter any one place or any one time and have a perspective on what's going on in the global ocean. So we do a lot of work to pull that all together. So we're involved. When I say we, it's Craig Carlson and I are making measurements. We share the responsibilities for measurements in the GoShip project. And this is the distribution of sections. The US does probably 2/3 or so of these, and we reoccupy a line every 10 years. And, the primary motivation for that work is to look at the invasion of anthropogenic heat into the ocean and the anthropogenic CO2 invasion into the ocean. And, I'm fortunate to be able to make measurements with my favorite variables as well. Right now, we have 2 cruises going on, one over here in this sector, and they're working their way south now, and then there's one over here, and these guys just blasted south, and they came across a big storm down there, and so this is the storm they were in. This is significant sea wave height. And it's about 10 meters height here. And this is where the ship was holding up for several days. And we're getting emails from friends on the ship suffering out there. And I'm certainly glad that I was not one of them. But eventually it passed by and they continued south and were able to reach the, the ice shelf down there, which kind of is disturbing to me because, they're not an icebreaker. So they're able to go through pretty clear waters, so that shows the effect of the warming going on down there. But anyway, so from those surveys, we get global distributions and inventories. And then there's wide ranges of ages of organic matter out in the ocean. And so C14 is used for that. We'll just take a real quick look at that. And then, once it's out there, we want to know, well, what controlled its production and what allows it to accumulate to higher concentrations where we see that. So we'll just touch on that. It's redistributed or distributed by circulation, and then it can be exported to great ocean depths, and that's typically in the North Atlantic deep water formation. I'll just touch on the microbial dynamics. There's a lot of dissolved organic carbon going through microbial sinks, and there's probably some abiotic sinks that I'll touch on as well. And then I just want to touch on a tracer for microbial removal of organic matter in the deep ocean, which might be relevant in other world oceans, just to introduce you to it. Okay, so how do we do it? The ship, here's a line. This is P16. This thing would be occupied over 3 different cruises. It's 14,000 kilometers, and all of our work is in summer, so it's always good weather work. This is the kind of distribution of sampling just in the upper 1,000 meters. We sample all the way to the bottom, of course, and we have these CTD rosettes that take water. Obviously it's got sensors for sampling on the way down for anything that you can have sensors with, but anything that you can't, that you need wet chemistry or anything like that, biological measurements, then you need these waters brought up. And so that's how we sample. And so when we put our data together and plot it out, we get a sense for what the distribution looks like. And what we see, of course, is surface enrichment, because, photosynthetic world out there. We see these nice streaks of enrichment, and those are indications of particles settling out of the water column and leaving a trace of dissolved organic carbon behind, which is, of course, supporting the deeper microbes here in the upper mesopelagic. And we are using this Shimazu analyzer. It's a TOC analyzer. And when we started this business, it was, we built these instruments ourselves. And we did hand injections with a syringe, and so it's now it's automated. We can let it run overnight, which is a wonderful advance for us. We have 3 of those instruments in our lab because we process a lot of samples from these cruises. It takes on the order of 6 to 8 months to complete one of these. One of these 3 sections here that if we occupy it, it's going to be 6 to 8 months before we have our data out. And then how does that system work? It's just the sample being acidified. We need to remove the inorganic carbon, so we lower the pH. We inject it with oxygen carrier into a high temperature combustion tube. CO2 is generated with just a very small volume required, 100 microliters is what we use. And then we have to dehumidify and strip the halides to protect the infrared detector at the end. And it's really that simple. It's simple, but it takes a lot of expertise to do it well. So the people doing the work in the labs do these work on these instruments just about every day. And so they always can tell when there's a little bit of something off, and they can make adjustments. So the quality is highest if you make the measurements very regularly. So if you go out and do a bunch of these surveys or these lines, then you can start mapping out global distributions. And so this is 3 lines or sections. Here's the Atlantic, the Indian, and there's the Pacific. And I'm accentuating the upper 600 meters because that's where the newly produced material is up here, the greens and the reds. And then I have 1,000 to 6,000 meters down here at depth. And the white lines are just showing us some selected density isopycnals, and the black lines give us a general sense of overturning circulation in the system. And so again, we see all this accumulation at depth. We see much lower concentrations at we see high concentration surface, lower concentrations at depth. And this stuff is at depth has a great age and a great, the bulk of the concentration out there. So it's about 640 petagrams of material that is, think of these pinks and blues as making up that material. And it has ages, in the bulk measurements that are several thousand years, 4,500, 5,000, 6,000, that sort of thing. So it's the old material that gets circulated with the overturning circulation. And then on top of that, when the water comes to the surface, then the phytoplankton and all the biological processes going on are generating some more organic carbon, and it can accumulate. So here in one gyre and here in another gyre, we see enhanced concentrations there, and here in the same thing in the Atlantic. And then if water can escape to the north, in the Atlantic in particular, then it gets caught up in the overturning circulation. So you see these enriched concentrations at depth here that we don't see in other ocean basins. And then it gets carried into the depths and then through the bathypelagic and basopelagic parts of the ocean, where you can see that this concentration gradient, it's blues and greens here, and it ends up as pink over here. So, but that's a very slow process of removal. It's over a time frame of 1,500, 2,000 years, something like that, where you have that kind of a loss. And I'll talk more about those losses in a little bit. Okay, so let's talk about how much carbon is being processed through DOC on an annual basis. So, phytoplankton are, of course, an important source of these dissolved components. Something bites into them, cytoplasm goes into solution. Do they get ripped up? Some zooplankton, defecates and lets some some dissolved components into the water. So that's why it's surface enriched. And it's on the order of half of all the primary production out there in the ocean is being consumed, through as it goes through the DOC pool, and the microbes are then consuming that. So the microbes have a massive role in consuming most of the dissolved organic matter, and it's really fast, right? So it's hard to see any of this stuff accumulating. So if you work in the world of microbial oceanography, you're very interested in this organic matter because it's the substrate for the microbes. If you're more of a biogeochemist, then perhaps you're interested in the material that isn't consumed as readily. And so there's about 2 petagrams of carbon per year on the order of 4% of global prime production that can, that we see, those enrichments up there in the surface. And that is the material that, if it finds its way to an area for deep ventilation, then it's going to be exported to depth, and there's going to be some loss of that material and convert it to CO2 and likely some other sinks in the deep ocean. So this is sort of the bigger picture of how that system works and the amounts. So let's think about the DOC accumulation. We make measurements out here in Bermuda as part of the Bermuda Atlantic Time Series study, BATS, and been doing that since the early 90s. And this is what the system looks like. It's just sort of a climatological look. Here's our cool winter. Here's our warm stratified summer. And during the winter, we have convective overturn, and that brings up nutrients to the surface waters, and that causes a little spring bloom. So here's our chlorophyll. We have a little spurt of productivity, enhanced productivity as a consequence of that. And then, of course, the nutrients are drawn down, the water's more stratified, and so then we just have a deep chlorophyll maximum here. So the inorganic carbon, that is, that's the material that is going to become our products, whether it's dissolved organic carbon, particular organic carbon, sinking flux, export production, that sort of thing. So it's mixed up with the nutrients, of course, and it's enriched at depth because that's where the mineralization products are, and then it gets mixed up, and then it's being consumed with time, and it's degassing because the waters are warming, but there's 30 to 40% of this loss of inorganic carbon that is biological. And while that is happening, dissolved organic carbon is one of the products of the net community production in the system, right? So that is, again, the 3 products of net community production would be DOC accumulation, POC accumulation, and POC exported as sinking particles. Okay, so that is what's going on globally. In this site, it's about 10% of the net community production that is found in newly produced DOC. And so if you want to go find where most of the new DOC is being produced, then you're going to go to the upwelling systems like off of Benguela, off of Peru, California, all those places, equatorial Pacific, anywhere you have new nutrients coming up the euphotic zone, that's where you have the highest net community production. And it is on the order of 15, 20% in those locations that accumulates as DOC, and they can be sent off into the interior of the ocean. All right, so here's surface DOC distribution. Let me just check my time here. And this is model distribution. Rainer Schlitzer did, and the background is the model, and these are our observations some time ago. And what we see is the enhanced concentrations in the lower and mid latitudes. And this is also where you'd find the highest temperatures in the ocean, right? And so it's a consequence of vertical stratification that allows this enhancement of DOC concentration. It's not being produced necessarily up in this area, for example. It's that the system is stratified by the main thermocline, right? The main thermocline holds warm waters and DOC-enriched waters up and prevents, deeper vertical mixing, and so you can keep enhanced concentrations. But if that water gets moved up to higher latitudes, so that it gets beyond the range of the main thermocline, such as in the North Atlantic, then it can, start mixing vertically. So we get these lower concentrations, these greens up here, and eventually it gets lost in the overturn. And some hundreds of years later, it's going to come under all of this and then pop up again in the Southern Ocean, and it'll have lost some of its concentration by microbial consumption of that DOC. So all the deep waters, the Pacific, Indian, and Atlantic Oceans, are coming back into the Southern Ocean in this way. They're all relatively impoverished of DOC. So that's why we get this big distribution in the Southern Ocean of relatively low DOC. So in the Atlantic, it goes down with some enhancement, but then by the time it pops up again down here, it's a lot of that material that the net export has been lost. Okay, so let me just, where are we? These three lines here, that one, and that one, we'll just look at those more closely here. So we're in the North Atlantic, here's North America, South America, Africa, Europe, etc. And here's that easternmost line, that is A16, this is A22, and this is A20. And so you can see that surface enrichment that we've seen in the other sections in the lower values at depth. And so we take that surface water, we move it up to the higher latitudes where I put a drain here. This is where North Atlantic deep water is going to form. And then we return it in the deep western boundary currents. And so, this green here is part of the deep, the exported material, the Iceland, Scotland overflow water, deep western boundary current, and it comes back around here. So you can see some loss of that material as it's being swept out. And then we can still see some remnants of that enhanced DOC in the South Atlantic here, and it's getting pulled up into the Southern Ocean because it's a nice divergence zone down there. So it's very nice to be able to trace this material all the way down there. So here we are at 3,000 meters. So we're really enriched up here because that's where most of the DOC is going to be exported to any great depth. And that deep circulation is going to kind of take this kind of a path, get caught up in the circumpolar waters, and some's going to head up here. And you can see a big global gradient concentration as well. And so what's going on here? We're, adding carbon to depth, and here we're sending it globally at depth, but then there are sinks in this location and in this location, and probably other locations. These are the ones that we can observe, all right? So these are local sinks that, there's DOC being removed there, and there's DOC being removed there. So it's not necessarily a slow biological removal of this material over the course of 1,500, 2,000 years. This stuff, I think, is mostly conservatively transported, but you create these sinks and then you mix. So if you take this water and this water and mix them, you get the gradient that we see here. But you have to have a sink up here to create that end member. So let's take a look at those. So this is, the deep Pacific, it's 4,500 meters. So this is salinity. And so we see salinity-rich waters from a large North Atlantic signal there. And the water's coming up here. It's getting a little fresher up to the north. And that's from overlying intermediate waters with the freshness. And so here we see this section here we're going to look at. And here's the high salinity water working its way in. Here's a little freshness from the intermediate water, so it becomes less dense by becoming fresher. And we also add some geothermal heat down here, so we get Pacific deep water returning. So if we look at the DOC distribution, it's enriched down here in the Southern Ocean, and then there's a gradient, and then we have this low value here and a much lower value here. And we can, do some estimates based on what we expect, to be there if it was all conserved versus what is there. And that allows us to determine how much DOC is removed in these two locations. So it's on the order of, highs of 4 micromolar or so in these two locations. So there it is again. So what's the mechanism for those? Because I don't think those are biological. The processes are way too slow. I mean, it's really slow. It's, 1,000-year type time frame. So I think they're abiotic. And so one would be, possibly slow scavenging of DOC by hydrothermal vent release particles. And let's just take a look at some of Jessica Fitzsimmons' work. Here's some geotraces crews, and there's the East Pacific Rise there. And, the gyre circulation is going to be sweeping across that area like that. And so here's our excess helium-3 coming into the system and being carried with that circulation. And when we look at these data, this is a north-south section, this is east-west section. So this section is coming right at us on this red line. Okay, so we're crossing this plume on this section. And here's particulate iron, for example, particulate manganese coming out and residing in that plume until they eventually sink out as particles. And so, Chris, I know he's online, put this nice figure together. So organic carbon, whether particulate in solution, is in, this rendering, binding, with iron and then sinking out. This is going to be a very slow process, right, as far as its impact on the DOC concentrations. But I think that it could go a long way to explaining these big deficits because we have a lot of time. Right? There is a lot of time. It's not something you can do experimentally to resolve. Okay. So another abiotic sink mechanism, this is an image from Susan Lang's figure. She wrote a chapter for the DOM book that I showed earlier, is just recharge of these hydrothermal systems. And you can see the deep sea water that goes in this 36 is a kind of value we'd expect to see in the Pacific, and 45 might be closer to what we'd see in the Atlantic. And you can see that it is then coming back out at much lower concentration. So there's a net loss there. Now, it could be that all of this material is being removed and then other organic carbon is being added through biological processing within the crust itself, because all of this material, as far as I recall, is radiochemically dead, radiocarbon dead, I mean, and this isn't. This has got, say a 5,000, 6,000 year age, and it comes out with, being dead. So it could be that everything is removed in the process, and then the microbes down here are adding more material to make up for it. But still, there is a net loss, and it could be that this is labile material to the microbes too. I don't know that. I don't know the outcome of that question. So if I project to an other world, and this is again, I don't have any experience thinking about this, but I found this figure and I saw that Fair is, I think, using this figure as well. And think about what might be going on in dissolved organic matter in particular. Well, if we have, autotrophs, then chemoautotrophy is playing a role, probably near the vents. And if they're down there, let's assume that there are some heterotrophs that developed as well. So we have that going on. But on Earth, at least, there doesn't seem to be 100% efficiency for the heterotrophs to break down the organic matter produced by the autotrophs, right? We always have DOM accumulate, we have soil organic matter accumulate, we have, fossil fuels accumulate, all this sort of product that seems to be inaccessible. So there's always a residue, And we wonder, why does DOM accumulate in the Earth's ocean? There's a lot of different molecular structure to these things. So it could be individually the concentrations are just too low, or maybe the enzymatic machinery to go after them is just too expensive. So microbes on the Earth tend to go after the material that they're more familiar with, biochemically, and less going after something that is very, 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 very diverse in its structure, right? So it does tend to accumulate. If it accumulates here, it probably accumulates elsewhere. And so how do you remove it, right? It's not going to just keep going up. So scavenging, if it's happening on the Earth, and likely would be happening in a hydrothermal system in another ocean, and the recharge mechanism as well, right? So here's our source term, and unless this just keeps going up and up and up, which doesn't seem like nature, then there are removal mechanisms that bring this to some happy concentration that's sort of at an equilibrium. So the final thing I want to touch on is another product that is interesting, is fluorescent dissolved organic matter. So we have DOM accumulate, but this DOM can be just a product of autotrophy, right? It's just something in the cytoplasm was not attacked. It was changed to some structure, wasn't attracted. But the metabolic processing of organic matter itself leads to the accumulation of fluorescent organic matter. And so, there are folks in the ocean sciences searching out the distribution of this fluorescent dissolved organic matter. So it's a tracer for metabolic, catabolic processes, and particularly microbial catabolism out there. And we can take a look and see what it looks like in the Pacific. We'll look up here, and the fluorescence was done over here, and oxygen from over here. So that's why I have all these lines here. But here's our oxygen. It's the deficit in the oxygen, so it's how much has been utilized. It's on the order of 300 micromoles per kilogram, and so this massive oxygen minimum zone here. And here's the fluorescence that accumulates as a consequence of the catabolic processing of that carbon. And it got a nice correlation to it. So there's fluorescence versus oxygen utilized. And all of these reds are in the abyssal waters, okay? And the blues are mesopelagic in the North Pacific. So it's a nice relationship. So the question is, would this happen in an anoxic world, right? Because this is oxygen utilized. What if we're in an anoxic world? So we can go to the Black Sea, and here we use H2S accumulation to get at apparent carbon mineralized, and it's CDOM rather than fluorescence, just another optical property of the DLM out there. And you can see there is still a very nice relationship between the growth of this optical property of DOM and the carbon that has been mineralized. So maybe, I don't know why it'd be in other world's oceans, but it'd be cool if it was. Why we just... All right, so thank you for your attention. I've got a lightning storm out in the back here. Am I still on? I hope so.
Yeah, we saw that. That was exciting.
Yeah.
Thank you so much, Dennis. I'd like to get to a couple questions. So let's see. Are there any observed anthropogenic contributions to DOC? Is that sort of identifiable?
Let me say, I work in the open ocean. If I can see land, I'm not very interested. So you'd have to, obviously be right up close in land. Some people are chasing down to the microplastics, break down enough to contribute to DOM. So people are looking at that kind of question. I think it's probably inevitable that if you're in a highly industrial estuary, you're going to see anthropogenic signal. It's going to go out. Some of it's going to be photooxidized. Some's going to survive. It's not my expertise though, so I can't add much to it.
Yeah. And I guess you were starting to get at this at the end, but do you ever complement the DOC measurements with compound or functional group-specific analyses?
Well, there's a big community of organic geochemists who are interested in that sort of thing. So you'll have to read the book to find out who they are. Buy the book and then read it. We have Liz Kuzhawinski at Woods Hole, and Krista Longnecker at Woods Hole. They wrote the chapter on metabolomics, so that's a new chapter in the book. And Dan Rapet up at Woods Hole wrote a chapter on organic matter composition.
Okay, we'll take one more, just following up on that. Do you know what compounds are contributing to the fluorescence of the FDOM?
I don't. There's a chapter in the new book on optical properties of dissolved organic matter. My understanding is that it's nanomolar kind of concentrations.
Yeah.
And so, you know, as a carbon biogeochemist, I'm not really interested, you know, in something that small. You know, it's just, you know, it's cool, but it just, to me, it's just a little optical property. You know, if I want to know how much catabolism took place, I'm going to look at how much oxygen was used as a biogeochemist, right? So I haven't looked personally at it, but you know, there are people who've loved that stuff, and that, you know, you'll find them in the chapter, having written the chapter and cited in the chapter.
Okay, and last question from me. So this was fascinating. In an abiotic world, does the DOC go way down because over geologic time, or is it at some steady state?
In an abiotic world?
Yeah.
How do you tell me how you're producing the DOC first?
That's right. I don't know. It was seeded there during the formation. This is probably a deeper discussion, but
yeah. Well, the lost terms, you know, if it was abiotically created, then it would still be, you know, if it's active with hydrothermals, then those sinks would still exist. In the ocean, you know, we lose it from photolysis in the surface, right? But presumably, you'd have to be in not a thick ice covered ocean for that to be going on as well. So,
okay, thank you.
All
Thanks
right.
a lot, Dennis.
I'll stop sharing then, right?
Okay, yes. Okay. All right, now let's move on to the next half of our talk with Dr. Fernanda Mora from JPL. She is a technologist and a scientist at JPL, working in the chemical analysis and life detection group. And her research is focusing on in situ organic chemical analysis with a focus, of course, on life detection, as well as, you know, the end-to-end methodologies for the extraction and processing of the samples to allow this organic chemistry. And so, Fernanda, please take it away.
Thank you, Aaron. Can you see my slides now?
Yes.
Okay, so thank you for the introduction, and I think it's going to be a little complementary to the previous talk because I'm going to talk about liquid analysis, so we can analyze those samples that you get, Dennis, with our instruments. Mostly I'm going to focus on the method portion of the work that we do at JPL. So very briefly, because I think everybody connected here knows that we are very interested in going to ocean worlds in the next, I don't know, decades, Europa and Enceladus, because they have liquid water and all of the elements that you will need for life to evolve. So we really want to go to these places and do analysis and try to find evidence of life. So how do we look for evidence of life? There are different approaches to this. In our group, we focus on life detection at the molecular level. This means when you look at life on Earth, life selects certain molecules for all of the processes that we need for life. It doesn't use everything that is available. But when you have an abiotic sample, you have statistical distributions of organic molecules, for example. So if you look at the abundance of a certain molecule versus a property of that molecule, in an abiotic sample, you don't see like a preference for certain molecules. In a biotic sample, you will get like spikes. It's almost like a fingerprint of what life needs. So this was described many, many years ago. We're just following. This is nothing new, right? Two very clear examples of these distributions that we understand well are fatty acids and amino acids. So if we look at amino acids first, amino acids are everywhere. We know that they have they can be produced both biotically and abiotically. But when you compare the distributions, you can see differences. So for example, if you look at the distribution of amino acids in meteorites versus the distribution of amino acids on a cell, on a microorganism, you can see that we have more complex amino acids in the biological sample, and the overall distribution is different between the two samples. So this is one way that if you look at the amino acids, you determine each amino acid on your sample, and you do these comparisons, you can tell if the sample has a biological component. The other very strong biosignature derived from amino acids is chirality, because mostly on Earth we use L amino acids and not so much the D amino acids. And we know in abiotic samples there is no preference, so you mostly end up with a mixture, 50-50 mixture of L and D. So this is a second biosignature that we get from amino acid analysis. And then for fatty acids, we look at the length of the carbon change on the amino acid. So if you look at the reaction that is abiotically produced, you see you form pretty much every length of carboxy of fatty acid. There is no preference. But when you look at biological samples, we know that the fatty acids have to be a certain length to give the properties of the membrane for the cells. So then you have these distributions. And we also, in biology, we create the fatty acids by adding 2 carbons at a time. So you tend to see more of the even numbers of carbons in the fatty acids than compared to the abiotic example. So these are only 2 examples of the molecules that we were interested in, because these are very clear patterns. But when you look at life, there are many other compounds that we use for life on Earth. So ideally, you want to be able to analyze as many of these things as possible, right? If you take cells and you start breaking them apart, I don't know if you can see my mouse, there's a point. If we start breaking them apart, you go from cell walls to lipids to fatty acids to carbohydrates. The same thing with DNA and proteins, etc. And there are other even inorganic ions that we use for life that will be interesting to analyze. So in order to decide how to analyze all of these molecules, one thing that we do is we look at we plotted the heat of vaporization versus solubility in water from some of these groups from the previous slide. And if you put here the heat of vaporization that you need for water, then you can start deciding whether or not you need to do, it's better to do a liquid analysis or a gas analysis. So the molecules that volatilize very easily and are normally not, they don't like water so much, they're better suited for gas analysis. Molecules that are more polar, like amino acids, and they are very soluble in water, they're very suited for liquid analysis. And I don't want to upset anybody. I'm not saying you cannot do this analysis in gas phase too, but the natural way is to do the liquid analysis if you have a sample that is already liquid and you're looking for polar compounds. So in our group, this is what we do. We focus on liquid analysis. And we focus on doing separation science because in order to find those biosignatures I just showed you, you need to be able to identify and quantify each of the species in your sample. So then you can calculate those distributions. So in order to do that, you need to separate the species so you can quantify them. And we focus specifically on capillary electrophoresis, which covers, you can cover a lot of these molecules on this space with this technique, sorry. So why do we do capillary electrophoresis? This is a very simple technique, just very briefly. All we need is a capillary tube, 50 microns, typically it's glass tube, it's empty. And what we do is we fill this tube with what we call the background electrolyte. It's basically a conductive solution. And depending on what we put on the solution is the molecules that we're going to be separating when we do our separation. So we fill this capillary with liquid, and then we inject a little bit of sample on one end of the capillary, typically nanoliters of sample. And then we apply voltage from between the 2 ends of the capillary. And what happens Next. What happens is that the molecules start migrating towards the end of the capillary, and they get separated based on their size and their charge and the interaction with the media, the liquid that we have inside the capillary. And then at the end, we always have a detector system, and what we get looks something like this. We get peaks that represent each compound. So based on what time we get a peak and the area of that peak, we can identify the species and we can calculate how much of that molecule was in the original sample. So some of the reasons why electrophoresis is good is because it's very simple. You don't need high pressure or packed columns. So it will be, why is not, this is not. It will be easy to implement on a mission. You don't require a lot of power. The hardware is very simple. We only need very small volumes of sample. I said that we normally inject nanoliters of sample, but we need microliters basically to make the instrument work, and probably low hundreds of microliters for the only instrument to work of reagents. So it's easy to miniaturize it. And the most important thing for this topic is also that we can couple it to multiple detection systems. So it's not restricted to one detector. We can do many different detectors. And in our group, we actually do 3 detectors, which is what I'm going to show you today. So I'm part of the chemical analysis and light detection group, and I try to summarize everything we do in one slide. And we basically do method development. This means we look at what are the molecules that we're trying to analyze, what is the best way to separate them, what method, a method that gives you the best sensitivity for those molecules, target molecules. At the same time, we also develop instruments that can do those analyses. And then in both cases, we validate the performance of the method and the instruments by analyzing natural samples. So we do standard samples in the lab, and then we do natural samples too. So today, what I'm going to show you is mostly one type of natural sample, but before I get there. So the instrument that we're developing in our group now, we call it Oceans, very clever name for analysis at ocean worlds. So it's the organic capillary electrophoresis analysis system. And very briefly, it has multiple components. So we take a sample. In some cases, we do some processes of the sample that include some extraction. We basically heat the sample a little bit. We need to do, in some cases, we need to do a reaction to do detection of the analytes in the sample. So that's all included like on the front end of the instrument. And I'm not gonna discuss a lot of this today, but I just wanted to show the whole flow. And then we do separation. So when your sample is inside the instrument, we can do CE, which is capillary electrophoresis. And as I said, we use 3 detectors. So the first one that we talk a lot is laser-induced fluorescence. And with this method, we can do analysis, chiral analysis of amino acids. And this is a method that gives us the best sensitivity, the lowest limit of detection. And then we have 2 more systems, 2 more detectors. Contactless conductivity, which is a very simple detector, and I will show you later. With this one, we can do inorganic ions, in addition to all of the some of the organic molecules too. The sensitivity is not as good as fluorescence, but it gives you a lot of information about the sample and the environment of the sample. And then finally, we also do mass spectrometry. And with this one, we have methods, again, for fatty acids and amino acids, and then some other positively charged organics like nucleobases and nucleosides that are derived from DNA. And we can also, in theory, detect unknowns. So if we are thinking about life that as we don't know it, then maybe with mass spectrometry, you can find new molecules that you were not expecting. And again, the sensitivity of mass spectrometry is not as good as laser-induced fluorescence, but for some of these methods, it's still pretty good. So with this, we cover pretty much a lot of the plot that I showed you at the beginning of like the chemical space, right? So this is what we do. We develop both methods and hardware. A lot of the samples, a lot of the work that we do, we have validated by analyzing samples from Monolake, which is is considered an analog for ocean worlds. It has a high pH and is very salty. So it also allows you to test the robustness of your instrument and your method when you have a sample that is not as clean and nice as the ones you prepare in the lab. So I'm going to show you the methods that we develop and how we test the results that we got with this natural sample. So as I said, we always start with the fluorescence method, because this one is the one that allows us the lowest sensitivity. This method is the most capable of the methods that are published with capillary electrophoresis for chiral analysis. We can do we can separate 13 amino acids, and actually it's longer, it's more that we can do now. We're gonna write another paper, but what is published is we can do 5 chiral pairs plus 3 non-chiral amino acids, and this covers the most abundant amino acids either on biology or found in meteorites. So we look at the most abundant and we can separate them and we can detect them at low nanomolar levels. So this is extremely capable, and we are expanding the compounds that we can analyze with this method, and we're gonna report that soon. So we use this method to analyze samples from Mono Lake, and what you can see here is we didn't need to do any pre-treatment of the sample other than adding the fluorescent dye that we use for fluorescence detection and a little bit of buffer to adjust the pH. So we normally do a 1-to-1 dilution with our labeling buffer. And we detected amino acids between 10 and 200 nanomolar. And then we did the same comparison. We calculated the distributions, like I showed you at the beginning, when we look at how does a meteorite look, how does E. coli look, which we know is biological, and then we look at Mono Lake, and you get very similar distribution to a biological sample instead of a meteorite. So this was the first biosignature. And then we also calculated the non-enantiomeric excess for the amino acids that we detected. And you can see that for most of the amino acids, it's 100%, so it's mostly L. But it's important to mention that there are exceptions to this rule of mostly L amino acids. And you can see it very clear here with the alanine signal. And the reason for this is because a lot of bacteria, they use the alanine on the cell walls. So that's where the alanine comes from. But this is still a biological sample, so it doesn't mean it's not biological, but it also highlights the need to do separation science and to measure multiple molecules, because if you only measure one thing, you cannot be absolutely sure that it was either biological or abiotic, right? So that's the bottom line. So moving on to the next very well understood fatty acid biosignatures. For this one, we developed a method coupling to the mass spectrometer. And just a brief reminder is where the fatty acids come from. They form the lipids that are part of all of our cells. And basically, they have a polar head and a non-polar tail. And so we refer to them, these numbers that you see here is actually the number of carbons on the tail. So we developed this method where we do first a reaction so we can detect these molecules with positive ionization on the mass spectrometer. So we basically add a group on the head that has a positive charge. And then you can see here, they're all separated. In this case, you don't need the separation to be perfect because you're also doing like a second level of separation on the mass spec because you are detecting only certain masses for the molecule. So you can see here, we go from 4 carbons to 22. And then we also did some fatty acid with double bonds. And you can see here that they can also be separated. So, and the LODs are very good. They're not 1 nanomolar like LIF, but they're still low nanomolar level. So this is very impressive for CEMS. And again, we use this method. Oh no, I didn't put the data. I put it there later. We also use it for the Mono Lake samples, and I will show you in a minute. The other method that we have for MS analysis is even simpler because it doesn't require any derivatization. We just take the sample and we can put it through the system and we can detect all of these compounds. And in this case, we selected a mixture of amino acids, also nucleosides and nucleobases derived from DNA, and some peptides, just to show broad analysis of molecules that we know we can detect with positive ionization mode. And this is the separation we get because we have 25 compounds. It's kind of hard to show it in one figure, so I had to make 2 figures, but they all get, they're all very well separated. This uses acetic acid as the liquid that goes inside the capillary. Again, it doesn't require any reaction or pre-concentration or desalting for this analysis. The limits of detection depend a lot on the compound and how well it ionizes. So they range from 50 nanomolar to 1 micromolar, which is, I think, very similar or lower than what you can get with other detection methods. And we validated this method also with high concentration samples containing high concentrations of salts, and we could still detect all the compounds in the mixture at the micromolar level using 3 molar sodium chloride. So very impressive. And then this is how the data looks like when you look at a natural sample. So on the left, this is the sample without doing any derivatization or anything. We detected mostly amino acids and some nucleobases. And then on the right-hand side is the monolake sample after doing the fatty acid analysis. And what you can see here is that it's not, when you have a natural sample, when you have multiple organisms living there, it's not so clear that you only see like 2 or 3 fatty acids, like the picture I showed at the beginning. It's a little bit more complex, but you can tease apart here which ones are the most abundant, and you can relate this to the type of organism that lives, that is living in your natural sample. And then finally, conductivity detection. This is the one that we use for mostly for inorganic ions and organic acids. We have 2 methods in this case. The advantage of this technique is that you can detect anything that has a charge because it's a conductivity detector. So it's really well suited for capillary electrophoresis because we're separating based on charge. And the other advantage of this technique is that all you need is 2 electrodes outside of the capillary. So this means that you can put this detector together with another detector in the same capillary. In this case, this is the same solution that I use for CEMS analysis, and you can get all of the fast-moving molecules are the inorganic ions, sodium, potassium, ammonia, and then you can also detect the amino acids with the same solution. So this will give you very relevant information about your sample if you can look at the inorganic ions first. And then we have another method focusing on the anions. And this one, we have like 20-something compounds, and we have more in this case that are not on this figure, but the same thing happens. Like early on the separation, you get the inorganic ions. In this case, we can do chloride, nitrate, sulfate. And then later, we can do relevant carboxylic acids that are relevant to metabolism, for example. And then we can do short chain fatty acids, not like the ones we did with the MS method, but some of these have been found in meteorites, for example. So this is also give us a broad detection with a single method. And again, the sensitivity is in the micromolar range, so it's not so bad. And again, we validated these 2 methods with the monolake sample. What you can see here is that because the sample has so much ions, you see a huge peak for the inorganic ions, and then you really have to zoom in to see the organic ions. To quantify sometimes the inorganics, we have to even dilute the sample even further because you saturate the detector. And the same happens with the inorganics, chloride and sulfate. And then I wanted to point out that we can also detect phosphate, which everybody gets very excited about. And it was recently reported in the plumes of Enceladus, and it's a very important molecule for life. So the fact that we can do this molecule complements really well the other analysis that we can do. Very briefly, I'm not going to talk about hardware today, but I wanted to show you that we have actual pictures and we do hardware. So all of these methods have been incorporated into our hardware at JPL. These are older versions of the instrument. These are more newer versions. We also, I didn't talk about it today, but Aaron actually has a project doing ion selective electrodes, which also complements very well all of the type of analysis that we can do. So the point will be to get the most possible chemical information of a sample with a single instrument by combining all of these detectors. So summary, I hope I show you today all of the things that we can do with capillary electrophoresis with a single instrument, simply changing the liquid that we put inside the capillary. We can do different detectors and we can cover a wide range of molecules related to life and habitability. As I said, yeah, we have 3 different detectors and we tested these instruments at Mono Lake, and we're ready now to be on a mission, hopefully soon. And I think with that, I want to thank the group, and I hope I didn't go so much over time, and you for listening.
Thank you, Fernanda. Let me bring up our questions. Let's see. Quick question is, any particular reason to use only capillary electrophoresis for analysis? Can you use other techniques like NOR or mass spec as well?
So we do, say it again?
I think, yeah, so what about other techniques like NMR or mass spec? I guess you can, mass spec is part
so
of.
we do, yeah, we do mass spec. NMR, I think the sensitivity is not as good as the as what we are doing now, and it doesn't allow you to do like the separation. We want to do separation and then be able to quantify each of these species. So that's why we do capillary electrophoresis.
Okay. How long can you use the same capillary until it's clogged by salt? Are there ways to remove salt from a capillary?
Yes, with acid. No, we use the capillaries for like months and months. It really depends on what we're doing. Normally, we don't see them clogging really. If we have a sample that we think might give us problems, we filter the sample before analysis, but we don't really see a lot of clogging. The capillary that we use for mass spectrometry is a little smaller. But again, we can filter the sample, we can filter all of the solutions that we use, and we don't really have issues with clogging. But if you do have an issue with clogging, you can always rinse it with ACL, for example, and it should dissolve whatever you have there. If you didn't dry it properly, for example, and you get like a salt crystal, you can do it with acid.
And here's one that's maybe for both Fernanda and Dennis. Is the fraction of DOC in the form of amino acids on Earth known, and is it relatively constant?
I'm gonna let Dennis take that one.
Yeah, yeah, people can make measurements of amino acids, right? And then they say what fraction of the total DOM pool is it? So that can be known. And it will not be a constant because if you're in the euphotic zone, there's a lot of production and release up there. And if you're down deep, then there's a lot of consumption going on. So it's going to have gradients in concentration.
Great. Well, thank you both. We're at the hour, and I do want to be conscious of everyone's time here. But there was one question that came in late, Dennis, for you, so another chance to go for the book. So, but is there another book that discusses particulate organic carbon? Do you have a recommendation or a link for that subject?
Nope.
Okay, well, if one comes to you, pass it along, and we can pass it all, to the now community as well.
Yeah, I can't. There's my marine microbial books and DOL biogeochemistry books, but I can't think of any particulate books. Because I think when you get into the particulates, it's more about life, right? So it's going to be productivity and all that sort of thing. It's not. I haven't seen a book about detrital material, for example.
All right. Well, thank you both again. Those were great talks. It was really great to have you.
Thank you for the invitation.